Well, good afternoon. I'm Kevin Murray. I'm the VP of Global Sales for Biospherics. Biospherics is an upstate New York company, and 30 years ago we developed the first controller to add oxygen-controlled incubators. And essentially since that day, we've developed multiple controllers and chambers to allow researchers to culture and process their cells in physiologic or pathophysiologic ways. Today I want to talk about a platform we've developed for the production of cell therapies. We like to consider it a better manufacturing practice. And, you know, most of you are doing all this great research, and ultimately one day you're going to get to the point where you're going to have to produce your cell product in a GMP-compliant way. And what is GMP? Essentially, GMP is quality. You need to produce a product that's free of contaminants, obviously, but also you need a consistent product each and every time you produce it, which is more difficult in the cell world because cells are alive and they react to their environment. So we've developed what we call a cytocentric barrier isolator. It obviously protects the cells, keeps them in a completely aseptic environment, isolates them from the technicians, but it protects the technicians as well. You know, you may be producing a cell product or a gene therapy that uses viruses and vectors and things of that nature where you don't want the technicians exposed. We allow to optimize the cell environment for whatever cell type you have. And we utilize process analytic technology and quality by design, which allows you to produce a higher quality cell product and have the data to show that. So I'm going to talk about a couple of things today: uninterruptible conditions, dynamic incubation options, and then cost savings. So what are uninterruptible conditions? You know, obviously the cells aren't exposed to any suboptimal conditions. You're going to get a higher cell quality product. It's a more consistent product. You get to continuously monitor and control the cell production from start to finish. There's no variations in temperature, pH, O2, CO2, which results when you take your cells out of the incubation environment to go work on them elsewhere. This is your clean room environment, of course. You know, you have your incubators, open hoods, maybe bench tops with microscopes or centrifuges or things of that nature. And what we do, and in the GMP world, you have to have this in a class 10,000 space, okay? We enclose and incorporate incubation with closed optimized chambers. And you only need a class 100,000 space, and that comes into the last section of my talk, which is cost savings. So typically, here's what happens in a clean room. You know, at night, everybody goes home, and you're optimizing for temperature, CO2, pH, and or oxygen, depending on what your protocol is. But when people come into the lab, they access the incubator, all of those conditions become suboptimal. I just stepped outside during lunch, and I started to shiver. You know, so you're basically, it's the same thing you're doing to your cells. They're in 37 degrees C. When you take them out, you put them on a cold surface, a bench, you're in a much cooler environment. The cells react, just like we do when we go in from a warm environment to a cold environment. That leads to variability, which is what you don't want when you're producing a cell therapy. In our system, we optimize the environment outside the incubation chamber to match whatever you're incubating those cells in. So you see no variation whether you're in the incubator or you're outside processing those cells. Excuse me. This is the same thing that happens when you take your cells out under the hood to go do some work. All those conditions change. They become suboptimal. And again, you're adding variability to your ultimate product. We open into, you know, processing is done in an optimized aseptic workspace, so there's no changes whether you're in the incubator or outside the incubator. So this is kind of a look at what typically happens. You know, in the clean room, you know, you're wearing the full bunny suits, aseptic garb, which isn't altogether comfortable, but there's still a possibility of contaminant getting into your product because there's not isolation. You know, our system completely isolates the technicians from the cells. If this technician right here sneezed, those cells would be fine. You could do anything inside that you typically do outside in a clean room. The difference is that whether you're in the incubator or outside here, nothing changes. Your O2 stays the same, your CO2, your temperature, All of those conditions stay the same whether you're in or out of the incubator. We even heat the surface to 37 degrees C, so if you put the cells down, they're not going to get any shock from a change in temperature. This is just a few slides on what the regulatory agencies feel. They like isolation concepts. The regulators want safety. So complete isolation of them is something that they're absolutely for, because you're dramatically reducing the chance of contamination getting in those cells if you remove the biggest contaminant in the room, which is humans. This is out of the EMA. Basically, isolator technology, they allowed for a system like this to go in a grade D room. And again, we'll get into why that's important later. So the systems run off 21 CFR Part 11 compliant software. But what you get to do is you can control and optimize a variety of parameters from O2, CO2, humidity, VOCs, particles, and get a real-time reading about what's going on in each module. You can set upper and lower limits so you get alarms if things go out of variation. You'll be aware of it. And this is quality by design. Essentially you'll know from start to finish what conditions your cells saw, whether they're in the incubator or out of the incubator, and you get a data log of that. So you basically have a batch record at the end of your production run to show that each and every time you made that cell product, it was done the same way under the same conditions. We also have a unique feature of dynamic incubation options. Cells are dynamic in nature, and the incubators need to basically adjust and keep up with those conditions. And this is very unique to our system. Let's say you're doing a cell expansion, and your gas phase has just stayed the same throughout the expansion. O2 being one of them, of course, as the cells are expanding, they're consuming the oxygen in the dish, and ultimately they become hypoxic. You basically counter this right now by splitting the cultures every so often and putting in fresh media and things of that nature. Our system can automatically ramp up the oxygen in the incubator so the O2 level in the dish remains the same during that expansion. You can do things like hypoxic preconditioning. One of the problems that many researchers have is they produce the cells, they put them in the animal model or whatnot, and most of the cells die. And a lot of that is mainly due to the fact that the cells adjust to where they're grown and processed and cultured. Typically, in most labs, it's done in room air. So you're taking cells that are gotten used to 21% oxygen environment, you've expanded them, you've grown them, you've isolated them, and then you stick them into an extremely low oxygen environment. It could be 0.5 or less. And right there you get a lot of cell shock, and a lot of those cells die. And there's a lot of literature that comes out, or has come out, about the benefits of preconditioning those cells prior to injecting them. This is just basically a little search with the terms hypoxia and preconditioning. There's more and more literature coming out that supports what this does. I'm not going to go through each and every one of these, but if you'd like the list, I can give it to you later. But essentially, in this one, MSCs gain a higher therapeutic capacity under hypoxia. Hypoxia preconditioning significantly improves capillary density and cell survival. And this goes on and on and on. The bottom line is maybe you wouldn't have to produce so many cells just to get enough of them to survive and have them graft and grow and do what you want them to do. The system's a modular design. This is actually a system that's at Roswell Park. They're doing a phase 1 clinical trial. It's a dendritic cell-derived cancer vaccine. They're producing the cells in our system here. It's modular, as I said. It's made up of basic components. You have a laminar flow hood, typical to a laminar flow in any lab. You're going to use it to basically prepare things that you're bringing in. You're going to debag them. You're going to wipe them down, things of that nature. Then you're going to go into what we call a buffer chamber. It's essentially an airlock or an air shower. And basically all outside air is then removed via dilution factoring, a log reduction, before you open the inner door. So you remove any outside air prior to opening the inside door. And then in the processing chambers, which is basically the rest of this schematic, We maintain a Class 100 or ISO 5 environment inside throughout. We use absolute HEPA filtration. We cycle the atmosphere inside about 70 changes per hour. And you control pressure, particles, VOCs, O2s, CO2, temperature. The only thing we change outside the incubation environment is humidity. There's a reason for it. If you happen to bring in a contaminant, they like warm, moist environments. So in a drier environment, if you happen to bring something in, it's likely going to get washed up through the HEPA filters. So these are just some images here. These are 2 large incubators. They're independently controlled, independently monitored. You could have different patient types. You could have different cell types. They could even have different conditions. You know, these could be at 20% O2. This could be at 5% O2. And prior to accessing the incubator, you just match the environment out here to whatever you're incubating those cells in. Basically, you can have any type of incubator, large, small, third-party incubators. Third-party equipment, you know, we specialize in a lot of custom builds. So no matter what you use, whether cell sorters, automated robotics, microscopes, anything in your production process, we can build a chamber, surround it, and keep it aseptic. This is actually a robotic liquid handler that we did for a project in Ohio. So you might see this is a wave bioreactor. Okay, why would I want this? Well, a lot of times there's upstream and downstream processing prior to putting things in a bioreactor. So essentially this particular customer wanted to incorporate the whole process inside their environment for their production. Just another examples of different things that we can and already have housed for various customers. And then the ultimate thing is why. If you are successful, you all know how funding is, and you're going to have to fund this. So you've got a couple options. You have a clean room option, which is what people typically build and use. But there's a lot of ongoing costs and hassles of clean rooms. Our system allows you to start small with what you need now and scale up when you do, should you need to. You can change your protocol if you have changes and you improve and enhance your methods. You can reconfigure the system to incorporate that. And it offers security in case funding dries up. Clean rooms are expensive. The operating costs are things that people don't think about very often. They get them built, and then they go, okay, we're ready to go. But at least here in the U.S., it's variable throughout the world. The operating costs can be extensive. HVAC, to maintain that grade A environment in a large room requires a lot of HVAC. And the energy cost associated with it is extremely high, at least, again, here in the U.S. Cleaning, again, clean rooms need to be cleaned top to bottom, ceiling to floor on a regular basis. In an isolator, you're not bringing in any bioburden. You essentially only have to wipe down probable risk surfaces, which would be the surface and anything that the person touched. You don't have to swab and wipe and clean the whole thing. So this cost is quite big. Even the garb, those garbs are one-time use. And depending on where you are, again, they could be $30, $40 a piece. You got 4 or 5 technicians. They come in and out of the clean room 4 or 5 times a day. I mean, that could be over $100,000 a year. People don't think about that. You don't need to wear the aseptic garb with our system. So, let's say the system that you purchased was the same as if you built a clean room. This is really what happens every year. Operating a clean room can cost $100,000, $200,000, $300,000 a year just to maintain. Ours is essentially about $10,000 to $15,000 a year. So this is where, in the long run, obviously, there's a huge savings, which you can use that money to grow, expand, fund more research. If you have a therapeutic product that's approved, your cost of goods goes down. So all of these good things happen. We typically speak with the end user about their process. That's the number one thing. How much incubation space do you need? How many people do you need working at the same time? What type of third-party instruments do you need involved with your production process? And based on that, we start to configure a system that fits the process, the protocol, the equipment, and the lab space. So they don't have to be linear all the time. They can obviously go around corners to fit the space that you're in. So you could begin to produce clinical grade cells in a dirty lab, refine your process over time, and then your transition into the GMP world is quite simple and fast. All you really need to do is take that system and put it in a class 100,000 space, which you can do really with a soft wall clean zone, and you're ready to go. So in terms of, the difference between our system and a clean room, we can be up and running in basically months. From the time we configure a system, we agree on that configuration, build it, get it to your lab, validate it. You're talking months instead of years. You have full-time data logging, which you can't do in a clean room. You can't possibly generate the data that you do in our system in an open clean room. You get real-time particle monitoring and limit control. You can expand. You can change, reconfigure. They need the full aseptic garb. We're both ISO 5. Decontamination, people ask, essentially the daily thing is just wiping down probable risk surfaces. If you ever got a contaminant, because I get asked this question, so I'm going to answer it now. We recommend a chlorine dioxide gas. If you have a confirmed contaminant in the system, it only takes about 8 hours. It's a gas, so it gets in every nook and cranny, and it's a 100% kill rate. So that's how we go about that. But you only do that when you have a confirmed contaminant in there. You'll still do environmental monitoring just like you do in an open clean room. You just do it in a smaller space. Currently, this needs to be updated, but we have systems all over the world. The blue ones, we do have a research system, which doesn't have all the bells and whistles that you need for GMP, but they can be added later if you ever get there. And we've gone through many of the regulatory bodies in multiple countries. They like our system. They like the isolation. They like the process control that you get. And that is that. So I kept it nice and quick and short and sweet. Thank you very much. Any questions? Well, if you're around, you want to hear more about it, I'll be around the rest of the day and tonight. And I appreciate your...
Purity level. Purity level.
I'm glad you asked. We don't use any outside air. It's all clinical grade gas. It's triple filtered on the way in, and then we balance the pressure inside with a vacuum, and that's filtered on the way out. So that would be one of the consumables is, O2, CO2, and nitrogen. The other are the filters. You change the filters on a semi-regular basis. But again, the cost of doing that, maintaining that, is far, far less than trying to maintain and operate a clean room. Yeah.
So your focus was on growing cells.
Mm-hmm.
Sorry. And do you also consider production of biological products?
We are actually getting into that now. I mean, we've been in the stem cell niche, really, or the area for probably 12 years now, but the biologics are now becoming more important. And in that world, a lot of the isolation is more important as well, depending on what you're culturing, growing, producing. So as of about 4 months ago, we started to target BSL facilities to see what their needs might be and how it might fit into their world. Yeah.
So with biological products, you refer products coming from living cells?
Mm-hmm. Yes.
I speak of biochemical
production
of
well,
products.
I'm not going to say no because we never say no. I was taught by my boss to not say no. But we haven't gone down that path yet, no. Mm-hmm. But if you want to discuss it, we certainly could.
Yeah. Okay. Yeah.
Sure.
Interesting. Thank you.
Yeah. Thank you. Okay. Just get that out of there.